Next time you get a chance, take a minute to look at the wide range of eye color among the students in your class. Chances are you'll find they range from brown to blue to green. You may also notice different shades and intensities of all these colors. Why? What causes these horses to have coats with such varied colors? What causes different blood types in people? And what factors are responsible for giving this cat its calico-colored fur? During the next few minutes, we're going to try to answer these questions and many others as we explore genetics in action. But first, let's review some of the work of Gregor Mendel, the father of genetics. As you may already know, Gregor Mendel was an Austrian monk who experimented with pea plants in his garden in the 1860s. He made many discoveries, establishing the foundation for the study of genetics. Genetics is the study of heredity, the passing on of traits from parents to offspring. From his work with pea plants, Mendel hypothesized that each trait has two factors. Today, we call these factors genes. Genes are the basic unit of heredity, carrying information about traits from parents to offspring. Each organism has two copies of each gene, one copy from the female and the other copy from the male parent. The two copies may have different forms. We call different forms of a gene alleles. For example, in pea plants, there's an allele that causes plants to grow tall and another allele that causes plants to grow short. The allele for growing tall is commonly represented by the capital letter T, and the allele for growing short by the small letter t. Mendel found that one allele often masks or dominates another. In pea plants, tallness is dominant over shortness. The allele for shortness, therefore, is recessive. While the concepts seem rather obvious today, at the time they were quite earth-shaking and did not reach their full impact until the turn of the 20th century. Another pea plant trait Mendel worked with was pea pod color. He found that green pea pods, symbolized by capital P, were dominant over yellow pea pods, symbolized by lowercase p. Mendel crossed a homozygous dominant green pea-podded plant with a homozygous recessive yellow pea-podded plant. All the offspring were heterozygous, and all had green pea pods. In other words, all the offspring had alleles in their gene pairs which were different from each other. You predict. What did the pea pods of these offspring look like? About 25% of the offspring were homozygous dominant, having green pea pods. About 50% were heterozygous with green pea pods, and about 25% of the offspring were homozygous recessive and had yellow pea pods. In these crosses, you can see that each parent contributes one gene for the trait to each offspring. When a parent plant forms sex cells, sperm or egg cells, the genes of the parent separate. Another word for separate is segregate. According to the law of segregation, a gene from each pair goes to each sex cell. For example, in the case of a heterozygous father, capital P and lowercase p separate to form sex cells. The same occurs in the mother. In the process of sexual reproduction, the male sex cell carrying a trait unites with the female sex cell carrying another trait to form a fertilized egg which eventually grows into a new plant. Each fertilized egg contains one gene from the mother and another gene from the father, for a specific trait. Gregor Mendel also crossed pea plants that differed from one another by more than one trait. For example, he made crosses in pea plants for the traits of both seed color and seed shape. He knew that yellow seed color was dominant over green, and that round shape was dominant over wrinkled. A special chart, called a Punnett Square, can be used to predict the possible genotypes of offspring for a single trait. We can also use a Punnett Square to keep track of possible genotypes in offspring where two different traits are involved. This chart is a little bigger, but it works the same way. In the first cross, he chose a parent that was homozygous dominant for both traits. This plant was bred with a plant that was homozygous recessive for both traits. The offspring of this cross outwardly displayed only the two dominant traits. In other words, the seeds were all yellow and smooth. These offspring were hybrids. Next, these hybrid offspring were allowed to self-pollinate. This type of cross is called a dihybrid cross because it involves two different hybrid traits. The offspring of the dihybrid cross revealed recessive traits for both characteristics. From these results, Mendel concluded that different traits are inherited independently of one another. This principle is known as the law of independent assortment. In other words, traits, or gene pairs, separate into sex cells randomly and independently of each other. In 1900, an important discovery was made by the German botanist Karl Correns. He found that the traditional principle of dominance and recessiveness did not always hold true. In his work with a type of flower called 4 o'clocks, he made a very interesting discovery. He crossed a purebred red 4 o'clock flower, symbolized by capital R, and a purebred white 4 o'clock flower, symbolized by capital W, W. You predict. What do you think the flowers looked like from this cross? Amazingly, these flowers were not red or white, but were pink. Correns hypothesized that in some gene pairs, the genes were neither recessive nor dominant. These genes are said to show incomplete dominance. Incomplete dominance may also occur in animals. When a purebred chestnut brown horse, symbolized by capital B, is bred with a purebred horse that is creamy white, symbolized by capital W, the offspring are a golden brown color. This type of horse is called a palomino and is the result of incomplete dominance. As you know, the human body contains blood, which you see when you cut yourself. Red blood cells, responsible for carrying oxygen throughout the body, have two possible carbohydrates which coat their surface. The letters A and B refer to these two different types of carbohydrates. A person that has blood type A has the type A carbohydrate in their blood cells, and a person that has blood type B has the type B carbohydrate in their blood cells. A person with type AB blood has both carbohydrates, and a person with type O blood has neither of these carbohydrates. Blood type is a codominant trait, meaning that each allele fully expresses itself. In other words, neither allele dominates the other, and each allele is equally emphasized. In most cases, a combination of many genes is responsible for a trait, as in the case for the trait of eye color. This is called polygenic inheritance. In polygenic inheritance, a group of genes act together to produce a trait. Most traits are the result of polygenic inheritance. In eye color, for example, different gene combinations produce an array of phenotypes. For this reason, there are a wide range of different shades of eye color. Height is another trait controlled by polygenic inheritance, as is skin color. It's estimated that 3 to 6 gene pairs control skin color, and it's estimated that even more gene pairs control hair color. Polygenic inheritance also plays a role in the traits of plants, such as in wheat, and in the traits of many animals. For example, egg production in chickens is a polygenic trait. About 30 years after Gregor Mendel conducted experiments in his garden, a Dutch botanist by the name of Hugo de Vries made additional observations on another type of plant called evening primroses. De Vries bred plants and got results similar to those of Mendel. But much to his amazement, every once in a while a new variety of primrose would appear. At the time, these unusual appearances could not be accounted for by the current laws of genetics. De Vries called the sudden changes he observed mutations. A mutation is a change in a gene. Mutations are rare events that produce new alleles in a population. When mutations do occur, they usually have no effect on an organism, or they may have a negative effect. As we just discussed, mutations can occur in plants and animals. They can also occur in people. Most of the time, mutations do not cause problems in the human body, but sometimes they can. Genetic disorder is the term which refers to problems in the body as a result of genetic factors. For example, sickle cell anemia is a serious blood disease caused by a genetic disorder. Normal red blood cells have a round shape. However, a person with sickle cell anemia has red blood cells that are misshapen. You observe. What's the shape of this cell? This cell is in the shape of a semicircle or half circle. These cells have a difficult time obtaining oxygen, making the person affected with this disorder weak. This disorder is the result of an individual inheriting 2 copies of the recessive allele for that trait. Sickle cell anemia exists mainly in people who can trace their ancestry to Africa. We now know that certain factors in the environment can also affect genetic disorders, such as harmful chemicals. And in some cases, ultraviolet radiation and sunlight may cause mutations in skin, leading to skin cancer. This is just a brief look at the very complex nature of some genetic disorders. During the past few minutes, we've explored some of the fascinating aspects of genetics. We reviewed some of the discoveries of Mendel, including the idea of dominant and recessive alleles. Each organism has 2 copies of each gene, 1 copy from the female and the other copy from the male parent. This is summarized in the law of segregation. Another law discussed, the law of independent assortment, states that different traits are inherited independently of one another. Later, we saw that the principle of dominance and recessiveness does not always hold true, as in the case with incomplete dominance. In codominance, we saw that neither allele dominates the other, and each allele is equally emphasized. We explored how combinations of genes can be responsible for a trait in polygenic inheritance. Eye color, height, and skin color are examples of polygenic traits. Finally, we discussed mutations, as well as a couple of genetic disorders found in humans. So, the next time you look in the mirror, observe a person's hair color, or notice the height of different people, think about some of the fundamentals of genetics we've just discussed. You might just look at your world a little differently. Fill in the correct word to complete the sentence. Good luck, and let's get started. Number 1. Genetics is the study of Number 2. Are the basic units of heredity. Number 3. A trait often masks a recessive trait. Number 4. An offspring, according to the law of segregation, generally receives a gene for a trait from each. Number 5. Different traits are inherited of one another. Number 6. The cross of these 4 o'clock flowers illustrates dominance. Number 7. Blood type is an example of Number 8. In inheritance, a group of genes together produces a trait. Number 9. A is a change in a gene or a chromosome. And number 10. Sickle cell anemia is the result of a disorder.